great to have an opportunity to tell you about some ongoing work here at the Quantitative Biosciences Institute. And we're going to try to highlight collaboration and in often cases how we're using CRISPR to facilitate that collaboration. So what I'd say here on the left is really the scientific vision of the Institute, and really my lab, is trying to put together different technologies in an integrative way and focus those technologies on a variety of different disease areas to get at the underlying biology across multiple disease areas to ultimately point in therapeutic direction. So we're heavily involved in combining mass spectrometry with CRISPR-based genetics, structural characterization, mostly cryo-EM, and chemical biology approaches woven with bioinformatics. And this pipeline is being focused at a number of disease areas in the context of these different mapping initiatives. So the cancer cell map initiative, the psychiatric cell map initiative there on the right, and then the host-pathogen mapping initiative. And we focused a lot on a variety of different pathogens. There I see Jacqueline is back. And we generate these protein-protein interaction maps, including HIV, which I'm going to speak about in just a minute, hepatitis B and C, different flaviviruses like dengue and Zika, and SARS-CoV-2, which I'll tell you about in a few slides. I'm going to let Jacqueline tell you, but a little bit more about the Quantitative Biosciences Institute first.
Yes, so we thought we'd give you a bit of a context of the Quantitative Biosciences Institute and how we've really set up a platform for collaboration and the various stories that we're going to tell you about. So the Quantitative Biosciences Institute, QBI, is comprised of 129 labs. The research is collaborative. It's quantitative, discovery science, disease agnostic, experimental, and computational. As Nevan mentioned, we're very well known for our cell mapping initiatives, which you see up here on the right. And however, we're very spoken about for investing in the young in science, and whether that comes in the form of creating, for example, the QBI Scholarship for Women from Developing Nations, or specific fellowships that take people from the beginning of their career into a platform, which we'll speak about shortly. However, we're most known for our events that are quite creative, and our international and joint symposia, which have been sort of the cornerstone of many of our collaborations around the world. And the QBI Fellowship, which I recently mentioned, is going to play a role later in what Nevan's going to talk about in the context of the QCRG, the QBI Coronavirus Research Group. The fellowship identifies promising standout candidates and speeds them up from graduate student to independent PI. And it's really a 2-year fellowship with an office space, a small lab space, and funding to be creative with. And this particular person actually has become quite a star in our COVID research at this time, and Nevan will talk a bit more about his research. We also started the QBI Scholarship for Women from Developing Nations in Biosciences, and really this is an opportunity not only for capacity building, but for really planting someone somewhere else to start collaborations across borders. And in this case, Jackie Lugemwa is from Uganda, and she's currently working with us on COVID as well as other initiatives of ours. The collaboration culture at home started the ground up, where we started with something called the QBI Happy Hour, where each month 3 labs nominate 1 postdoc that have 5 minutes and a maximum of 3 slides to share their work and start talking with beverages and hot food. And we found that there was a tremendous amount of collaborations that started within the campus in these 129 labs at that point. So as we mentioned before, we have a scientific vision of collaboration through the cell maps, but and Nevan talked about these maps, which he's going to dive into quite shortly. And we started thinking at the same time, well, what if? What if in the same way we map cells, we started mapping the world with collaborative interactions? What if cell mapping equaled world mapping? And what if protein-protein interactions, in fact, also equal people-people interactions? And what we found as we started mapping the world, we started looking at where we were going and what interactions we were creating with different people, institutes, academic or industry. And we started finding that, in fact, people started falling into very specific networks, much like the protein networks that we've been talking about. And people grouped around whether it was cancer or technology, infectious diseases, and such. And importantly for our upcoming story on the coronavirus research group, you'll notice that both New York and Paris were already in our infectious disease network. And at this time, we have a number of collaborations around the world and still growing. We sort of feel like the world is our oyster, I suppose. And so we have a number of places that we've been interacting with, ranging from Haiti to Uganda to China, and really very fruitful collaborations, especially in times such as the pandemic. And so the formation of the QCRG, the QBI Coronavirus Research Group, was really the understanding of the context of a coming pandemic and taking action. However, really, when we look back, how did it all start? It actually started with HIV research, which led the path for the SARS-CoV-2 research. And Nevan's going to tell you a little bit about this.
Right. So as Jacqueline said, the foundation was really laid. And I think that's an important point to make is all the research money that went into studying HIV and other viruses was so important as many virologists pivoted to study SARS-CoV-2. So as we said, we're in the business of generating protein-protein interaction maps. On the top left, the first map we generated with the pathogen was HIV. So we generated an HIV human protein-protein interaction map. We identified 435 human proteins that physically connected to at least one of the HIV proteins. And we wanted to do was to try to functionally validate those interactors. Did they have any effect on HIV infection? But Judd Hultquist, a postdoc in the lab who is now a professor at Northwestern, collaborated with the Alex Marson lab here at UCSF, and he set up a system where we could use CRISPR to knock out human genes one at a time here in primary T cells, the most relevant system for HIV. And here's the pipeline that he set up, and then we could knock these genes out and then infect the cells with HIV and see what effects the knockouts have. And what we wanted to do here was essentially not just knock out one gene, but knock out all 435 genes whose proteins we found physically connected to at least one HIV protein. So the goal here was to try to set up a high throughput, 96 well format, a pipeline in which we could do this. And Judd and Joe Hyatt, a student that works jointly with myself and Alex Marson, did just this, where we're taking multiple donors, using a CRISPR-Cas9 RNP system, getting that into cells, knocking out genes, infecting with HIV, GFP HIV, so we could look at monitor HIV infectivity. Did this at days 3, 5, and 7. And then importantly, we're also sequencing all of the cells to see how effective the CRISPR knockouts were, because we wanted to correlate infectivity with CRISPR knockout efficiency. So here's a picture of Joe late, it was like 3 a.m. one time with the picture that he took, that I think Judd took of him. And this just represents a relatively small slice of the reagents that went into this particular study. So as I said, we were sequencing all the cells that we tried to knock out so we could generate these type of graphs where each node corresponds to a gene. On the x-axis is editing efficiency as determined by sequencing. On the y-axis is the effect on HIV infectivity. So what we're interested in are the things in the top right-hand quadrant, genes that we know we could knock out that have an effect on HIV infection. So if we go back and look at our protein-protein interaction map, 435 human proteins, like I said, we tried to knock them all out. We got most of them knocked out. And 86 of them, when knocked out, had a statistically significant impact on HIV infection. Since it was quantitative, we could find dependency factors and restriction factors, genes that when you knocked out, infection got better. So in blue here are genes that are dependency factors. In red are restriction factors. And overall, we got about a 30% validation rate in this particular cell system, which is incredibly high. As a comparison, what we did was just an RNA-seq experiment, as many do, in the same cell system when you infect with HIV. We took the top 75 genes, upregulated the bottom 75 genes, downregulated, knocked them all out, and we got 1 out of 150 that had an effect. And in fact, it was the same one that was on our protein-protein interaction map. To me, this illustrates the power of looking at protein-protein interactions to find biology. So using this type of logic, we pivoted quickly when SARS-CoV-2 hit in the context of this QCRG that Jacqueline alluded to, the QBI coronavirus research group involving many different groups at UCSF. It involves many different academic institutions around the world and a number of different very exciting industry partnerships as well that we've been working with over the last year and a half. So we were the first group to clone out each one of these genes, the SARS-CoV-2 genes, about 30 of them. So what we wanted to do is, like we did with HIV, put affinity tags on them, purifying them from cells, and then look via mass spec, what are the co-purifying human proteins? And this is one of the first maps that we generated, carrying out this particular process. The red nodes are the viral proteins, the circles are the human proteins. So in total, we had 332, a list that we gave to Brian Shortcat and Kevin Shokat. And then they gave us a list of compounds and drugs that would bind to and inhibit these set of proteins, 69 in total, that we would hope would be enriched for the virus to be needed to infect our cells. So we collaborated with New York and Paris, as Jacqueline told you about, and found that many of these did have antiviral effects. Based on all our work, there's actually 26 drugs in clinical trial right as we speak being tested for COVID-19, 2 that we're really intimately involved in, 2 translational inhibitors, Aplidin and Zotadefin. Aplidin, which is also known as plitidepsin, is from the Spanish company PharmaMar. It's actually in a phase 3 clinical trial right now at 70 sites in 17 different countries. So we're very excited to see the results of these clinical trials, which will be made available very soon. Hopefully we'll have another tool here in the toolbox to fight COVID-19. So just to quickly segue to another paper, a very collaborative paper, as you can see, where we generated a protein-protein interaction map, not just to SARS-CoV-2, but SARS-CoV-1 and MERS, because we wanted to do cross-comparative studies. We also did localization experiments, combined it with CRISPR-based genetics, which I'm going to highlight here in a second, did also structural analysis, and also tried to connect this to real-world clinical data. So you can see there's 200 authors on this particular paper from 14 different institutions in 6 countries. I would say a work like this normally would take 6-7 years. It took about 4 or 5 months, and that's a testament to the collaborative spirit that went into this particular effort. So based on today's seminar here, I'm going to just jump to the CRISPR-based analysis that we did. So the genetic analysis, we took 332 interacting proteins, worked with Veronica and Marco there at the top at the Pasteur Institute, used RNAi to knock down these genes in A549 cells expressing ACE2. We used RT-PCR as our readout. In parallel, with the arrow going down to the bottom, we collaborated with Synthego, specifically Jared and Kevin, where they knocked out each one of these genes using CRISPR and CACA2 cells. We sent those cells to Chris Bassler. He did another assay for us where he used an antibody against the SARS-CoV-2 protein as the readout. So we had 2 different systems here that we're looking at. And then the bottom right, in total, out of the 332, we had 73 genes, at least in one of these systems, that had a statistically significant impact on HIV infection. Another way to look at this is in a network view. This is what I'm showing you here. The blue nodes are the 73 genes. The edges correspond to physical connections to the different coronaviral proteins. I want to zoom up on one part just in the middle here. It's a connection between ORF9b, a SARS-CoV-2 protein, and it exists in SARS-CoV-1, actually not in MERS, but we found it physically interacting with this protein TOM70, a mitochondrial protein. And we found that when you CRISPR out TOM70, you have an order of magnitude decrease of infection in CACA2 cells. So we had a number of lines of evidence suggesting that this particular connection was very important. And Klim Verba, who leads the QCRG structural biology group that Jacqueline told you about, the QBI fellow, he and his team got a cryo-EM structure of TOM70 binding to ORF9b. So ORF9b is in orange there, and TOM70 is in green. And TOM70 is doing a number of things, including regulating mitochondrial function and the immune response. We actually think it's the latter function that ORF9b is manipulating for the benefit for the virus. And we think that this is connected to a number of variants as well. Just on the right, there is some information about the structure. 36 out of the 38 ORF9b residues are interacting with TOM70. And there's a couple of phosphorylation sites that we identified on ORF9b that if we're phosphorylated, there's no way it could interact with TOM70. So we think phosphorylation is regulating this key interaction. And we're focusing a lot of attention on this interaction. And I love it because it shows you the value of combining proteomics and genetics and structural biology and virology to narrow in on a key node, which we think is important for SARS-CoV-1, SARS-CoV-2, and ultimately SARS-CoV-3. And we're doing a lot of fragment-based screening right now to find a compound that would interrupt this particular interaction. And as I alluded to before, we're generating a lot of information on these different variants. We think this is a key node that the alpha and the delta variant is using to better infect our cells. And we're going deeper into that. I won't talk to you about that, but I will say is that we're looking at proteomics, drug screening, CRISPR-based genetics, transcriptomic analysis, and also a work in bat cells to do a comparison across these different variants. And just very briefly, we use a similar approach across many disease areas. We had 3 papers that came out just a couple of weeks ago in Science. We're combining essentially proteomics and genetics, mostly CRISPR-based genetics, to get at insights behind a variety of cancers, including head and neck cancer and breast cancer, and working with Triadiker, putting all this information together to come up with a hierarchical model of the cancer cell, which we're now using in combination with AI to make predictions about precision medicine. So we're very excited about this platform, and we're going across many different cancers and many other disease areas as well.
And so we wanted to tell you also a lot about the future plans we have for collaborations using the various platforms and technology that we have that Nevan's just gone through. And we're currently focusing on collaborations in Africa that we have. Mainly, we are starting with the University of Ghana, where we are working on a couple of cancer projects with them. But they have high sequencing capabilities, which we're going to be using along with eventually CRISPR and proteomics. And we are with all these countries that we're going to show you, we are assisting them and working with them to get mass specs into the area. We are also working with Uganda. I mentioned earlier our scholar, Jackie Lugemwa, who's there, who's currently the only person in the country with knowledge of mass spectrometry, because during her year at UCSF, she took a course. And so we are reinforcing that relationship there, and we're currently working with her on COVID and HIV and looking at their abilities for sequencing, as well as further down the road to be doing some knockouts and various things with cohorts and samples that they'll be sending our way. We are also working with Nigeria, where they have actually the capacity to house various machines, such as cryo-EMs or mass spectrometers, because they have an institute of note that is almost finished. And we are working with Christian Happy there on various viruses, Lassa, Ebola, Marburg, to name a few. And finally, we are working with our partner in Sierra Leone, Byler Barry, who is currently also doing a Ph.D. in policy at UCSF, although he's already doctor of note in Sierra Leone, who opened a significant clinic on HIV, which was also a first responder to the Ebola outbreak there at the time. So we are looking at having a symposium in Sierra Leone this coming year. It's been postponed for 2 years now, which will be the first symposium on infectious diseases in Sierra Leone. And we invite any of you to reach out to us to be part of this. We are therefore looking at various future plans and breaking silos worldwide. We have a number of QBI scholars for women who have been lined up, but due to the pandemic held back, from Poland, Nigeria, Ghana, and other places. And we soon have a grant that we put in at NIH with 8 institutes in India and 6 in the United States for exchanges of trainees on infectious diseases such as tuberculosis. And the symposium, as I mentioned, sorry. And finally, we're planning on a psychiatry and neurology symposium with an institute in Argentina. And with this, we'll be wrapping up by telling you about a paper that Nevan and I were asked to put together for Cell to describe our work in creating collaborations around the world and what we use. And it's a bit of a how-to piece of how to involve young scientists, academia, industry, philanthropy, importantly, media and communications, and as well the use of symposia to create a dialogue around visions. Nevan?
Yeah, and just to add to what Jacqueline said, I think a lot of us would agree that there are problems with the scientific enterprise, academia and industry. And one of the major problems is that it's incredibly siloed, as Jacqueline alluded to, but it also rewards the individual. One person gets a grant, one person gets an award, one person gets tenure, and in doing so, you often discourage collaboration. But during this pandemic, it's been remarkable to see how fast we can all move together when we work together, when we break down these silos and don't worry so much about these individual awards. So I think the challenge now is to take what we've learned and apply it to the future. And the question now is, when the dust settles on this pandemic, are we going to keep this infrastructure and spirit alive so that we're better prepared for the next pandemic? And then the larger question is, why aren't we working together like this on all diseases, on breast cancer, on Alzheimer's, on Parkinson's, on heart disease? And I think because of this pandemic, which has obviously been a tragedy, there's a silver lining. One of the silver linings is in 20 years, we're going to look back and say, hey, what you learned during that pandemic, we applied to all these other disease areas, and we have so many more treatments, and we can help out so many more people. So that's the goal. And as Jacqueline said, her and I wrote up a piece describing what we did during the pandemic and how it can be used in the future. And actually Nature as well highlighted this collaborative effort. It was actually on the cover of Nature a couple of months ago. So if you're interested in these activities, what we've done and what we plan on doing, I'd encourage you to read these 2 pieces. And here's the last slide. This is a picture of our lab that was taken a couple months before the pandemic. There, obviously, a lot of people are involved in this, and just to highlight a few, the bottom left is Dave Gordon, the upper right is Wen Zhang. They spearheaded our protein-protein interaction work on SARS-CoV-2. Mehdi at the back there is a postdoc. He's a computational expert. He helped out in all of this work. And then Robin Cake-Ruth and Daniel Sweeney, the remaining people circled, fantastic mass spectrometrists. They're crucial for all of this work that they described and all the work that we really do. So I'd like to thank them, and we'd like to thank all of you when we get happy.
Thank you.
Hey, great. Thank you both for such an informative talk. There are several questions in the chat. If you wouldn't mind taking a moment just to answer one before we move on. One of the audience members wants to know, have you thought about doing proximity labeling?
Yes, we have actually developed approaches in the past using Apex, the Apex tag. We are doing this. We're also bio-ID and turbo-ID to get at, obviously, the more transient interactions that could be important. We're also employing cross-linking mass spec approaches to get at those transient interactions. But the one thing that I think I'd maybe end with saying here is, which is kind of related to this question, one of the things that we're doing now is combining CRISPR, mass spec, and cryo-EM all together by tagging on the genome with CRISPR important factors that are connected, host factors connected to, say, SARS-CoV-2 or HIV, purifying them, plus and minus infection. You can do this poorly. Doing mass spec, doing cross-linking mass spec, and going right to cryo-EM. Okay, and to me, that is incredibly exciting to temporally look at how these complexes change during infection and combine all these different approaches together. CRISPR, proteomics, and cryo-EM. So it's not just CRISPR, it's not just mass spec. It's about putting it all together and combining it with chemical biology and structural biology. That's the vision here, and I think that's where the big breakthroughs are going to come. And one of our visions here, obviously, as Jacqueline alluded to, is to try to get some of these technologies in the hands of people where some of these diseases are incredibly problematic. So it's about connecting genes and proteins, but it's also about connecting people around the world.